reactions and an amplification of the cell, the signals within the or intracellularly. So this is known as the signal transduction pathway. So this kind of signal transduction pathway or the cell signaling can be either of many types. So you have local, long distance, you have paracrine, justacrine, autocrine, that is within the cell itself. So we will come to that in a little bit. So when it comes to that, chemical messengers, usually hormones, if you can take them, and animals and plants, they have cell junctions. So you have CAMs, cell adhesive molecules, then you have desmosomes, and then you have junctions wherein the cells directly are connected to the other cell's cytoplasm. So local signaling, you have animal cells wherein, there's this direct contact through this cell adhesion junctions. They communicate with each other or by recognizing each cells. So when it comes to animal cells and plant cells, you have something called as in the plant cells, plasmodesmata, which helps in the cell signaling molecules to transfer within the next cell from one cell to another. In the animal cells, you have the cap junctions. So when it comes to local signaling in animal cells, it can be direct. So you have the cell that is being directly in contact with the next cell through a carrier protein or through the next protein that is present in fact, and signal molecules which help in attaching to the receptor on the next cell, and the cell signal is being passed on through signal transduction. In other cases, you have regulators. For example, in paracrine signaling, what happens is one particular cell, which is the secretory vesicle, for example, transfers the signaling molecules through a local regulator, or for example, a growth factor into the ECF, and this ECF goes and binds onto the proteins on the target cell. So this causes the target cell to act. So what can happen with that? It can either change your transport, it acts on the transport proteins, it acts on the cytoskeleton, it acts on the enzymes and as well as the genes. So this is the target finally. When it comes to synaptic signaling, that is in the nerves, your transmission of nerve impulses, what is happening is, it's again paracrine signaling because both the cells are adjacent to each other. One cyton and axon are both adjacent. And for example, again, nerve transmitter acetylcholine can be released. This neurotransmitter diffuses across the synapse or the synaptic junction and causes the signal transduction and the signal amplification to take place. Now we have long distance signaling. In this, we have both plant and animals using hormones. So your hormones are being released at one particular point. For example, your thyroid hormone, which is present at the base, of your throat. So from there, it has to go and act on each and every cell that is present. Your paracrine glands release your insulin, glucagon, and they are present in your pancreatic cells. They have to go, and these receptors are present on every cell within the body. So it's quite a distance from your your little toe to go for the insulin to go and act on. So how are they going to do? Travel through the blood vessels. This to the target cell finally. And then you have these specialized endocrine glands released into the lymphatic cells or the blood cells and will reach the bloods. So cell communication, not given this example. Anyway, who had discovered the cell communication basically? It was discovery of epinephrine hormone acting on the cells. So there are three signals, whatever cell that is receiving this particular hormone or signal will undergo three processes. One is reception, identification, that is there's an external or there is a signaling molecule that has come and is attached to onto the receptor. Then transduction. And then finally the response. Once this amplification or transduction has done, what is the response from that particular cell? What is it supposed to do? So when it comes to cell signaling, you can see usually you have a ligand that is present and a receptor. So this is the signaling molecule and this is the plasma membrane. extracellular fluid, cytoplasm. So again, once this is done, what is going to happen next? This signaling molecule should relay the information what it is carrying or why is it present to within the cell. Finally, it should go and act upon the targets, the target, which is what is the result that we are going to get. So you have a relay of molecules. So many are there in that signal, inside the transduction pathway molecules, in fact, or amplification molecules. So this causes a pathway to act. And then what will happen? So cellular response within the cell, what is it causing? Transcription, translation, or cell, what do you call it, apoptosis? Or is it going and acting on the CSK? Or is it again releasing other enzymes? So yes, you have, so this is, no, all these are known as, in the transduction, you have secondary messengers, tertiary messengers, which are going to act upon, causing what do you call it, a cellular response. So when a signal molecule binds to a receptor protein, then it causes a change in the shape. So there is a change in the target cell receptor so that, there is, upon recognition, the signal binds to a specific site. It's a receptor is a protein over here. So there is a change in the shape of it, or opening or closing of the channels, or the ligand which binds to causes the receptor protein will cause a change in the shape which activates the receptor. Now, we have quite a lot of receptors. There can be intracellular or and then you have different kinds of receptors also over here. When it comes to the intracellular receptors, again, small ligands are present, which are hydrophobic molecules. They can cross into the cell membrane also. So, one is example is testosterone. And some receptors that detect ligands are intracellular. They are found in cytosol or the nucleus of the target cells. That is one thing. And a receptor hormone complex will act as a transcription factor. And also you have hydrophobic messengers that example is the steroid and the thyroid hormones. What do? They pass through the plasma membrane because they can pass through this lipid bilayer and then binds to a receptor protein which is present in the cytoplasm itself. And then this causes the transcription, translation to occur and protein synthesis to occur. So this is what happens in steroid and thyroid, and if there are any kind of receptors in the plasma membrane, they are of three types again. G protein linked receptors, tyrosine kinases. What do? They phosphorylate, or an addition of phosphate. Kinase enzyme is that is the action of it. And you have channels. So when it comes to G protein coupled receptors, what exactly is happening over here? Again, these G protein receptors, they interact with proteins, which is known as G proteins. Towards the cytosolic side, they're present, and then they activate and cause a cellular response. So the signaling molecules, these ones, what are they? Estrogens, eicosanoids, or catecholamic signaling molecules. That is your dopamine, adrenaline, noradrenaline, all of these. They can also be peptide hormones. They can also be neurotransmitters, acetylcholine, glutamine, GABA, serotonin, all of these. And then neuropeptides as well. And you can have chemokines, interleukins, nucleosides, all of them can be a signaling molecule. Once they are there, what will happen? Again, you have different types of G protein receptors, in fact. So in this G protein receptor, you have what do you call it, exoplasmic domain, in fact, and then you have a transmembrane domain, and at the same time in the cytosolic region you have a cytosolic region of this G protein, which actually is going to interact with the G proteins. So that is why a G protein linked receptor is also known as a 7-TM receptors. We'll come back to that in some time probably. Uh, when it comes to receptor tyrosine kinases, these are basically your signaling molecules that are present, which are right. So there again enzymes. This signaling molecules itself is an enzyme, and these receptors have an enzymatic activity within them. And that is why they are also known as having intrinsic enzymatic activity. So this receptor tyrosine kinases have this intrinsic enzyme activity within their cytosolic region itself. You can just see over here. And these phosphorylate tyrosine residues. So with the help of signaling molecules such as adapter proteins. And then hence that is why it's known as a signaling what do you call it, receptor tyrosine kinase. It itself is a receptor, it itself is going to change. So you have and the signaling molecule can be your different growth factors, and it can be insulin or insulin like growth factor as well. So this is one thing about receptor tyrosine kinase. When there are two signaling molecules, they come form a dimer, activated tyrosine kinase. So phosphorylation takes place, and then release of ADP to activated ATP is released, and that the phosphate bond is broken down, and you have a fully activated receptor tyrosine kinase, which is along with the phosphate which is attached from the breaking of ATP. Once this is done, again, you have a cascade pathway causing a cellular response. And you have ion channel receptors, wherein what happens is, as soon as the ligand comes, signaling molecule, then it is opened, and it is usually the ion channel is closed. But with the presence of the ions, this ligand-gated ion channel receptor opens, and these ions go inside. For example, your calcium-gated channel, sodium-gated channels, they're all ion channel receptors. This causes cellular response, so there's a change inside the cell itself. So what happens in the signal transduction pathway? So molecular interactions are going to relay the signal. It's usually many steps together, and signal amplification is there. One molecule can, for example, your CAMP pathway, one molecule will go and activate many. That is why it's known as a cascade reaction, and transmit this signal to multiple molecules. This will in turn go and what do you call it, transmit the same signal to many more multiple molecules, resulting in a large number of molecules that have been activated with a specific function for the cell to actually give a response. So usually as a receptor, single signal molecule is going to usually be bound. This activates another protein inside, and activation of that is going to happen. So what is it that we are seeing in the case of phosphorylation or dephosphorylation? That is the basic signal that is being transmitted. Phosphorylation, dephosphorylation. So protein kinases, like I said, receptor tyrosine kinase is one. You have serine threonine kinases. You have histidine kinases or protein phosphatases or the guanylate cyclases. All of these. What happens is, in these, binding of the signaling molecule to the receptor. So you have the receptor dimerization and autophosphorylation to take place. And once the phosphate group is removed from the protein, it gets deactivated, dephosphorylation, and it's back to its inactive state. So this is what happens in protein phosphorylation and dephosphorylation. So because of which that signal has stopped, that reaction has stopped because no more phosphorylation has occurred. So in the phosphorylation activity or a cascade, as you can see, you get to see that it is a signal molecule. Like I told you, it can be any kind of signal molecule. For example, growth factors, or it can be what do you call it, your insulin or insulin-like growth factor. So this is the receptor. It can be tyrosine kinase or serine threonine kinase. Activate, it gets activated. Of course, energy is released over here. If at all it has to get phosphorylated, meaning that ATP or GTP has to break. So it's usually having an inactive protein kinase inside the cell, and this gets activated through phosphorylation. So relay molecule is also present at the same time, activates the protein kinase, with which what happens is this activated protein kinase transfers one phosphate group from the ATP to another inactive molecule of the another protein kinase. So in case if it's a MAP kinase, mitogen activated phosphate kinase. So what happens is that is getting activated, transfers another protein kinase. So to another kinase. So another inactive protein kinase to active protein kinase. Again, involvement of energy is present. And this active protein kinase 2 activates or catalyzes the phosphorylation of another protein kinase 3. So MAP kinase pathway, if at all I give you that example, that is how it is happening. And then you have a MAP kinase, and then MAP kinase. So it's in this manner. And finally, this active protein kinase 3 phosphorylates the cell's response through activating the inactive protein which is necessary to active protein involving in a cellular response. Whichever protein has lost the phosphorylation or phosphate group will become inactive again. So this is how you have your phosphorylation taking place and dephosphorylation taking place. You have one enzyme, second enzyme, which is a phosphatase, removing that phosphorylation, phosphate group. So you have small molecules, ions, they act as secondary messengers. So some molecules that include are secondary messengers are basically your G protein coupled receptors, receptor tyrosine kinases, and then you have CAMP pathway, calcium ions, sodium ions, all of them, inositol phosphate. So different kinds of secondary messenger system are there which are present intracellularly. So CAMP pathway is very important. We will have a look at it later. CAMP pathway is basically cyclic AMP. So what is happening is you have your ATP and that has only two phosphate bonds released, and it becomes a cyclic form of it which is known as adenylyl cyclase. So this is one important pathway that we get to see in some time. In fact, it is very important. And Sutherland, in fact, he was studying in the effect of epinephrine. He was actually studying about cyclic AMP. So the major component is your cyclic AMP signal transduction pathway. We will come to that. So adenylyl cyclase is the enzyme which converts the ATP, adenosine triphosphate, to CAMP in response to an extracellular signal. So you can just see this is ATP basically. In the presence of the enzyme, it loses two phosphate bonds and we have obtains a cyclic form. And whenever there is a phosphodiesterase being used, you have the ester bond, ester bond being cut or cleaved, and water molecule comes out. And this forms the AMP. But this cyclic AMP is very important and it is an extremely important cell signaling molecule within a body, within each cell for that matter. So, and phosphodiesterase helps in cleaving this bond. Diesterate, so two both ester bonds are cleaved. So you're going to get this particular signal. And then with the help of CAMP, when it comes, CAMP pathway actually goes and triggers, or whenever molecules trigger the formation and CAMP, this activates protein kinase A again, and with this protein kinase A inside the cell phosphorylates other proteins. So finally, thanks to CAMP pathway, its target within the cell is to cause extracellular response, the cellular response, which can be production of transport proteins or cytoskeletal components, enzymes or hormones, or it goes and acts on genes causing transcription, translation or cell death. So if you see a G protein coupled receptor over here for that matter, we will come back to CAMP because that is important. So GPCR, like I told you, it's a transmembrane protein, 7 transmembrane protein in fact, causes GTP and then finally it releases, it acts GTP, this activates the adenylyl cyclase, because of which again it forms this adenylyl cyclase. When it gets activated, then ATP is converted into CAMP, secondary messenger, which causes the release of protein kinase A and then cellular receptors responses. Now we have calcium ions and inositol triphosphate. They also act as secondary messengers because this calcium is important because the cell concentration can be regulated, and calcium again is used in many parts. For example, it is like, making an impact on your heart muscle contraction, your skeletal muscle contraction. Again, calcium ions play an important role in nerve impulses and so many other places where calcium is playing an important role. So, so you have different kinds of calcium pumps that are present on the mitochondria, on the endoplasmic reticulum, and all of it is being triggered by both high and low concentration of calcium. Now, this leads to release or involves another molecule which is known as diacylglycerol and IP3, which is inositol triphosphate, which are again secondary messengers which relay within the cell itself to, for other kinases or for other molecules to be triggered. So, what happens whenever DAG and inositol triphosphate are acting upon, this causes increase in the cellular calcium. So, for example, any signal molecule is present. So, this causes, in the case of DAG or calcium pathway, please give me a moment. In the calcium signaling pathway, let me just like, so you have different kinds of signaling molecules. So when it comes to that, DAG and IP3 are secondary messengers again. So whenever secondary messengers, which are kinases, they come over, you can have IP3, which is inositol 145 triphosphate, and DAG, which is diacyl glycerol. So they go act upon the cytosol. Again, release of or activation of phospholipase C is happening, because of which cleavage of phospholipid PIP2 is taking place, which is phosphatidyl inositol 4,5 triphosphate, and IP3, which is phosphatidyl inositol 3,4,5 triphosphate. So in three places you have phosphate groups present. This DAG, when it gets released, acts as a secondary messenger again causes release of IP3. This goes and acts on, whenever there is this IP3 coming upon, it goes acts on a calcium gated channel, because of which whenever there is extra calcium molecules in the endoplasmic reticulum of the same cell, then it releases the calcium from the endoplasmic reticulum into the into the cell again. We have learned last time that, ER has calcium deposits, or ER stores calcium. So this, within the cell also, for the calcium to be released into the cytosol, the signaling pathway works. Then IP3 diffuses through the cytosol, binds to IP3 gated calcium channel in the endoplasmic reticulum, causes the ER, causes it to open. So from the ER, you have calcium, cytosolic calcium levels increasing. This activates another protein through another signaling pathway. So what exactly happens? So because of this, you have release of protein kinase. Then these targets are again different kinds of proteins, enzymes, transcription factors. So whenever there are transcription factors that are actually getting activated, you have gene expression, that is growth, proliferation, differentiation, or you have cell death. So, like I told you, it is the cellular and the nuclear responses that we get to see. So, any kind of cytoplasmic responses, binding of epinephrine C. We are looking at your cyclic AMP pathway basically over here, CAMP pathway. So, epinephrine or no, is going adrenaline or epinephrine is binding to a G protein coupled receptor. And this inactive G protein coupled receptor becomes activated. How many molecules of that? Around 10 power 2, around 100 molecules of G protein coupled receptors. These 100 molecules each will activate another 100 molecules of adenylyl cyclates to form active adenylyl cyclates. So 100 into 100. So around 1 lakh adenylyl cyclic molecules are being, or around 10,000 of them are being activated. Now this CAFP is going to go and act upon inactive protein kinase A. This is again amplified 10 power 4 molecules at a time. That is around 1,000, 10,000 molecules at a time. We already have 10,000 over here, 10,000 into 10,000. Look at the kind of cascade of reaction, and it is an amplified signal over here. This inactive protein kinase A gets activated and this activated protein kinase A activates phosphorylase kinase. So again, release of them around 10 power 5 molecules over here. This goes, acts upon inactive glycogen phosphorylase, causes active glycogen phosphorylase to be released. So what is a glycogen phosphorylase going to do? Acts on glycogen and forms glucose 1 phosphate molecules. So glycogen phosphorylase is going to add a phosphate group to the glycogen, and then end of the day you're going to get glucose 1 phosphate. And then if it in this manner activates the the DNA growth factor, again your cascade signal amplification is taking place, gets into the nucleus. Here again inactive transcription factor gets activated. Your transcription factors are activated, because of which you have the DNA, and it's acting on the genes, because of which you're getting a gene expression. It can be growth, differentiation, or proliferation, or again a signal to cause cell death, because of which it goes proteins for the necessary of my apoptosis are being released, lysosomes are activated, and your BCL family too, and then your Bax family, your other family of genes are being activated, because of which apoptosis takes place. So again, we have two things that we have to remember, that is amplification of the signal and how much of it is being a necessity. So different cells, different proteins you have, you should prevent crosstalk to stop happening over here. So cell response can lead to a pathway leads to single response, or again two responses, and then you have if at all there's any activation of another cell, this causes another complete reaction to take place. So according to that, you have different kinds of proteins. Again, so you have scaffolding protein signal complexes which are going to relay the signal and see that it does not, there is not much of crosstalk taking place. So the signal molecule with the receptor on the plasma membrane acts on the scaffolding protein. You have different kinds of protein kinases. Only the necessity one should be activated upon, not all of them. And then termination is basically usually when the phosphate group, in case of phosphorylases, are cleaved by a phosphatase, or that group is being transferred to the other activate the inactive protein kinase, for example. Then what happens is the previous one, the phosphate donor, whatever is there, is automatically becoming inactive. So receptor, relay molecule, and activation of cellular response is what we are looking at. So please look for signal transduction pathway in the cell again, and you can have a look at it in white and white or lineager. You'll get good information regarding that. I'll have to just see if at all I've got. Just give me a moment. I wanted to talk about. So we'll just have a look at it as such. What is it that's causing? So response to light and heat and receptors. So external is mating factor, for example, and bacteria undergo quorum sensing. Long distance and short distance, we have already seen autocrine, endocrine, and local is paracrine and synaptic. And then I think I have just shared this particular uh, signaling molecules can be like ligand and molecules as well. And you have ion channels, G protein coupled receptors. Please give me a moment. This is not what I wanted to show you. This is done. This is also done. Okay. And of course, you have different kinds of receptors also that have been like, that get to be shown. So, yes, different receptors again are like, you have alpha beta receptors that are present, and then you have the cholinergic or muscarinic receptors. So, and you have what are they going to do? How are they going to work on contraction relaxation basically? So as the name says, so they are going to cause muscle relaxation, and adrenergic receptors stimulate the contraction of the heart muscle for that matter. And what are the components? Like I said, epinephrine or many signaling molecules, anything can be there. Receptor, effector is the cascade molecules that are present inside. These, for the receptor, the signaling molecules cause a change in the adenylyl cyclase, for example, phospholipase C, protein kinases A, B, C, whatever. And so these are the effector molecules. Now, this effector molecule helps in translation of the message to the secondary messengers, which are the CAMP, CGMP, cyclic adenosine monophosphate, cyclic guanosine monophosphate, inositol triphosphate, diacylglycerol, calcium. These are the secondary messengers. So, these secondary messengers again transfer the information or the message whenever they themselves are there inside the cell. So it causes effect on the muscle contraction, secretion, or any other like target for the necessity of your activation of action on cytoskeleton, gene expression, your protein synthesis, all of that for that matter. So this is a G protein coupled receptor in fact, and this causes it has alpha and beta gamma subunits. So this alpha subunit of the G protein, this is the receptor and this is the G protein. So in this alpha beta gamma is there. Whenever this alpha subunit goes and gets attached to the alpha GDP plus beta gamma subunits, and this causes the beta gamma subunit and alpha subunit to form. So alpha with the GTP will act on the effector. That itself is an effector again. This goes and this acts on the adenylyl cyclase, because of which CMP levels are increased. So again, what is an agonist and an antagonist? Agonist is a chemical molecule when present inside the system will help in increase of this particular action to take place. Antagonist is atenolol if it is if at all it is present. Then what happens is it is going to stop this or causes a decrease in the adenylyl cyclase, because of which increased CMP levels are not going to take place at all. So decrease the CMP levels. So you have different kinds of receptors. I'll send this particular PPT also if you want. So you can just have a look at it. So many types. Oh, sorry, I didn't share the screen at the first place. So you have downstream effectors which cause different kind of reactions altogether. Like I told you, protein kinase A. In case of muscles, causes contraction. If it is not present, then causes relaxation. And downstream effectors, again, if you see calcium, it acts on the calmodulin pathway, in fact. And whenever calcium in the smooth muscle causes the release of calmodulin, and when calcium calmodulin is present, it goes and acts upon which is known as something known as an MLC, which causes contraction. If it is not there anymore, this calcium calmodulin is not there, then the muscle is going to be relaxed. So you have adrenergic receptors also. Again, different types, causes different kinds of constriction as well as vasodilation. So both are again acting on your G proteins. Different subunits are present. So you have phenylephrine, prazosin, all of this being agonist and antagonist. Overall effect is again increase in the calcium levels inside the cell. So this is how your calcium calmodulin pathway is going to take place. So we have seen so far like the signaling pathway and the cell signal transduction. And now I'm going to stop my share. We have one more lesson which is gene regulation, or the genetic code and the protein synthesis, which I will start with. So this is all about your signal transduction pathway. So in IP3 or calcium pathway, just give me a moment. I will check whether we have IP3 also with us in our syllabus. Genetic code, structure, selection. CAM, inositol phosphate, we have prostaglandins. Most like they all act through the same way. So now what I'm going to talk about is the genetic code and the protein synthesis. Let me open the respective slides, please. So important questions, whatever is there behind your textbook, just please follow the previous questions. That is enough. You need not just go through what do you call it, the whatever questions are there, previous question papers, at least the last 2 3 years question papers, and the ones behind your textbook, whatever is given. If you follow that, that's more than enough. Right. I hope this answers your question. Wait, let me start sharing screen again with you. Cell signaling is reverse in nuclear instruction. So, right, genetic code, transcription, translation. So, what exactly is the genetic code, and how are we going to see this? Yeah, that's all right. Sure. So, when it comes to genetic code, what is it going to do? What is it that we are talking about when we talk about the genetic code? So, how are the nucleotides the present inside your genes? Genes are made up of nucleotides. And how are these nucleotides affecting your amino acids or proteins? So, how is a protein or a DNA formed in the first place? How is a DNA formed? And how is a protein formed with these, a set of DNA and RNA? So, it is the set of rules that is present in the DNA or the RNA sequences, which finally gives you the proteins necessary for what do you call it, the cell proliferation or cell to exist. So it was a term that was given by George Gamow, and it was discovered by again structure of DNA thanks to James Watson and Crick. Because of this, they found out the code which is going to be like on your amino acids. How are the amino acids present to give the respective proteins? So again, we are going to go for adenine, guanine, thymine, cytosine. They are the nucleotides present in DNA. Of course, we have uracil, we have other modified purines and pyrimidines. But then usually that's what you're going to get: adenine, guanine, thymine, cytosine. Adenine, guanine, double bond, guanine and cytosine, triple bond, and corresponding to they are coding with each other or complementary to each other: adenine, guanine, thymine, cytosine. So this are going to be organized into a codon. So finally, this forms a genetic code. So in this, what are we looking at? 20 amino acids. So it should be 20 codons. And each codon should have 3 nucleotides that are going to specify to that amino acid and to a specific codon again. So one nucleotide, you can have around 4 combinations. 2 nucleotides, you're going to get 16 combinations. So 3 nucleotides, 64. That is what am I talking about? So adenine, thymine, guanine, if they are present on one. And then yes, the DNA, it is double-stranded. You have adenine, guanine, thymine, cytosine. And on the other side, it is complementary, right? With the respective one. If there is adenine over here, there should be thymine over here. Guanine over here, cytosine over there. So they are complementary to each other. And this will be transcribed and then translated to form a respective protein. So when three nucleotides are present, they code for one amino acid. So that is what we are trying to tell. So again, this can be expressed in a simple table, and of this you can actually see whenever there is three nucleotides present, we have UCAG, uracil in the case of proteins. So adenine, guanine, thymine, cytosine, when transcribed, you're going to, after transcription of the DNA, you're going to get a basic, transcribed DNA, and this codes for the production of proteins. So after transcription, you're going to get a uracil instead of a thymine. So you're going to get UCAG. These are the four, genetic, the, what do you call it? The nucleotides that are present, which cause the production of, if it is UUU present, it is going to be phenylalanine that is going to be there. UUA, leucine. If it is CUU, or the first two letters are going to be the same, the last letter can be, or the last nucleotide can be again UC, that is cytosine, or adenine or guanine. It is still going to code for the production of a amino acid, that is leucine. AUU, AUC is going to be isoleucine. So this is how it is going to be in the second position again. In the third position, you're going to get UCAG, anything is going to cause the production. So in the first amino acid, first nucleotide can be UCAG, uracil, cytosine, adenine, guanine. In the second position, if it is again the same UCAG, it is going to have the same production of the phenylalanine or leucine. And if the third one is again UCAG, since it is a triplet and codon, we are going to get the same amino acid. So these genetic words are called as codons, triplets. They are 64 in number, and these, in the 64, three of them, UAA, UAG, and UGA, are known as stop codons. Whenever after translation, you're going to see these in the end. That means that protein synthesis, after, when it comes for after translation, whenever the during protein synthesis, this codon comes, that means the synthesis of that particular protein is going to be stopped. So they are known as stopped codons. So it is 61 codons for 20 amino acids, and 3 do not code for any amino acids. Again, what are the different types of codons? You have sense codons and antisense codons, missense codons, for example. So we will just see. We have sense codons, signal codons. And the sense codons code for different respective amino acids. Signal ones are those which will initiate, stop, or give that particular signal to either initiate the protein synthesis or stop the protein synthesis. What are they? Usually for us, it is methionine, which is AUG. In the case of bacteria or prokaryotes, it is formyl methionine. And then you have UA, UAG, and UGA, which is amber, ochre, and opal. So these are again the stop codons. And there are two types of signal codons, like I told you, termination and initiate. Amber, ochre, and opal. These are the ones. And initiation are AUG. And in the case of prokaryotes, it is formal methionine. Base sequence again. You have many kinds of mRNAs present, tRNAs present, rRNAs present, many kinds of RNAs that are present. So these code for, or the base sequence that is present in each pool of tRNA. One tRNA can have an anticodon. It can be having AGG. It can be having, what do you call it, um, UAG, no? So it is UAG. So it is AUG. So like in that manner, AUC. So whenever that is there, the transcription can start or translation. So in the mRNA, if it is AUG, so it is initiation of the protein synthesis. So over here, that respective tRNA which will come, which has the codon, which is UAUG, no, UAG. So this is a position in which this particular nucleotide can change. It's known as the wobble position. This is the tRNA having an anticodon loop. This is the mRNA having these codons present. So what is the difference between a codon and anticodon? You have to know that. So it can be present in both the DNA and RNA, and but the anticodon is always going to be in the tRNA position only, obviously, because it has to go attach itself to the mRNA or the DNA to cause the particular process, be it transcription or translation to occur. And again, you have directions over here, which is codon is always going to be in the 5 prime to 3 prime. And to read this, it should be complementary. So it should be in the opposite way, which is 3 prime to 5 prime. And anticodon always, some of them at least will pair with more than one codon. And they are arranged in the nucleic acid strand, whereas anticodons are present in the cells. They can be attached with different kinds of amino acids as well. Like I told you, important topics, or the materials that is, like, you can just have a look at the PowerPoints that I keep sending. I have sent the previous lessons once to Kishore sir. It is just for referral. Your textbook has got good information in it. You need not, at least for your exams, go for or beyond your textbook. And if you're preparing for competitive exams right now after writing your MSc, and if you want to qualify for your CSIR or what do you call it, or you're going to teach, then please refer Cell. Then please refer White and White. White and White has got very good biochemistry by White and White. I mean, Leninger, and so these are the books that I usually refer for biochemistry. So that is one thing, and that's what I hope this is good for your helps you. So coming back to our lesson, when it comes to your codon, based on the codon only, your anticodon comes and joins, so that protein synthesis acts is carried forward. So what is the characteristic of this particular genetic code? It is a triplet code, and it has not got any kind of pause in between, and this code is not going to be overlapped, and there is a particular way in the code it is going to be read, and it is degenerate, and it is universal for any living organism, be it right from your bacteria to human beings and any other living organism. There's an initiation code, and it is non-ambiguous. We are not going to fight upon is this going to be changed or not. And then you have a particular code which is AUG, which is going to be coding for the amino acid methionine. It is not going to change anywhere except for the fact that in the prokaryotes it is a formal methionine. But whenever there is a methionine amino acid present, it means that protein translation or the protein synthesis is going to take place in any organism. That is what they mean by chain initiation code. Termination codons again, UAA, UAG, UGA are not changing in any of the organism again. So the code is triplet. 64 codons are there. If you have a double, again, like, some, like I told you earlier, number of amino acids that can be formed are can be given basically, like I told you, one nucleotide, four combinations. So two nucleotides, 16 combinations, or 364. So what we mean is this: three nucleotides are present, and if one is there, four combinations are there. Again, if two nucleotides are present, UC, UG, UA, like, then you're going to get only around 16 combinations. But with the three nucleotides together, you're going to get 64, of which 61 are coding, and it is universal. So there is no change in this, except for, again, universal, but then in mitochondria it is methionine, but in AUG, in cytoplasm, it is going to be isoleucine. But within this, there is no other change except, maybe some exceptions are there. Like I said, F-met. Non-ambiguous, you do not get any other codon to specify another amino acid. Non-overlapping, meaning that only one base cannot participate in the formation of more than one codon. Continuous, there is no pause at all. It is always transcription, translation occurring simultaneously, starting at the start codon, stopping at the stop codons. Polarity, again, definite direction, 5 prime to 3 prime, and then amino acids coming from the 3 prime to 5 prime, and the reading of it will give you different amino acids if you see the mRNA from which end you are looking at. So UUG stands for leucine. If you read from right to left, that is your 5 prime to 3 prime. Or if you're going to read it from 3 prime to 5 prime, you're going to get a valine. So polarity is there. Degeneracy is any amino acid. The last amino acid, if it is changing, not a problem. But if you change the first two amino acids, then you get a different the first two nucleotides are changed. You're going to get a different amino acid altogether. So this is what you mean by degeneracy of the genetic code. Amino acid can be coded for more than one codon. And also at the same time, if both the first and second nucleotides are the same, the third nucleotide, even if it is different, it will still code for that respective amino acid only. But if the first two amino acids are changed, then the third one will code for a different amino acid. And Crick had postulated the wobble hypothesis, which shows that according to this, first two amino acids have, first two codons have a normal base pairing, but the third codon can change. Like I told you, if it is like, UCAG, any one of them can come into the tRNA, and then if it is there, it's causing a wobble pairing. So this explains the degeneracy. That is, even if the third codon is of a different nature, the same amino acid is being produced. So what is the biological significance of it? So according to this, you have around 20 codons that have been known for your amino acids, and 44 codons, the last ones, they cause the chain to get terminated, the protein chain to get terminated. So because of this, it is going to be higher, the what do you call it, chain termination. So how is the genetic information being flown, or it is seen? We have DNA in our cells, so this DNA has to get transcribed. So DNA, you have nucleotides. This causes the formation of mRNA. DNA is being transcribed to mRNA. This mRNA is being read by a process called as translation to cause or to have protein synthesis. Proteins are made up of peptides. So peptides are made up of amino acids. So proteins are basically made up of amino acids. So anyway, what is it that we are going to see? We have mutations, wherein what is happening is, if there is any kind of codon that is being changed, deleted, added, or the reading is changed, causes a mutation. Point mutations are silent, missense, nonsense. Frame shift mutations are your reading frame has shifted altogether, because of which you're going to read it in a different manner. You're going to read the next codon, because of which you're going to get a different amino acid, and the whole protein is going to change. So silent mutations are single nucleotide. For example, ATG codes for methionine. So GAA, glutamine. So in this case, whenever there is a silent mutation, it's the same amino acid, because the first two nucleotides are not changed. So this mutation is not going to make any difference as such. Missense mutation is when what is happening is adenine changes to cytosine. So different amino acid will come over here. See, adenine, cytosine. So what is happening is over here CAT. So you have this histidine. These are the amino acids that are going to come. Instead, what is happening is over here you have the replacement of CAT with CCT, cytosine, in the mRNA, because of which you have, instead of histidine being released, you have proline. So it is an incorrect amino acid. The protein is not going to function. Nonsense mutation, again, you have the production of a stop codon being generated. So prematurely, the protein synthesis is stopped, and the because of which you have a like incomplete protein to be formed. Frame shift is what is happening is, if at all there's a removal of a base, so the reading frame changes, because of which ATG, GAA in this manner. What happens is, if at all the G for guanine is removed, it is going to read as AAG, adenine, adenine, guanine. So this causes the production of a different protein or different amino acid, and this causes the production of a different protein altogether. So this is what we see in the genetic code. Now let us come to that factor of protein synthesis. Right. We have seen just the basics of like I um, request you all to I want to extend the class for another half an hour if there's a possibility, please, so that I'll be able to finish even gene regulation. So share my screen. Yes. Right. When it comes to protein synthesis, basic. We have looked at transcription. I think that was already done for you. Uh, when it comes to the protein synthesis, check the gene expression of regulation. I do not know whether transcription was done to you or not. Like, right. Mechanism of protein synthesis, or we are going to see basics of transcription translation in this initiation, that is starting of the the start codon. Basically, we'll just have a look at it. Genetic code, protein synthesis, uh, components of the protein synthesis, which is ribosomes in the eukaryotes, prokaryotes. This also I have dealt with in the earlier class when we spoke about ribosomes, different units. I'm going to pause my share uh, and just take and talk about ribosomes once. So we have different types of ribosomes that are present, right? Let me just share my screen. Yeah. Right. Ribonucleoproteins. Pallet was the first person to uh, see. And what are the types again? You have different types in different cells that are present. In fact, two types again, matrix ribosomes, plasma membrane ribosomes that are present. And they have two domains, that is translational domain, exit domain. And prokaryotic ribosomes are of 70S and they weigh around 2.7 daltons. They have two subunits, which is 50S and 30S. So when it comes to the structure, you have a larger subunit, which is of 50S, which is like S is Swedberg unit, and it has 23S, and then it is having 5S, that is 5 Swedberg unit, that is the weight basically, rRNAs, ribosomal RNAs. And then you have in the smaller subunit 16S rRNA, it is consisting of 21 proteins. So when it comes to that, I have already spoken about the Swedberg units. So when it comes to the smaller subunit, its molecular weight is around 0.9 into 10 power 6 daltons. It's made up of 16S rRNAs and 21 polypeptide chains. It has a head and a platform and a base. When it comes to the larger subunit, molecular weight is given and 5S rRNA, 23S rRNA, 34 polypeptide chains. It has a ridge shaped structure. It's like finger-like basically. It's got a front, it's got a stalk and a ridge. This is the back, how it is going to look from the back end. This is the front, this is the back, this is the ridge, and this is the central protuberance. Please imagine it in the 3D manner. So this is necessary for transcription translation. So we will come to that. RNA is transcribed from DNA, in fact, that is mRNA synthesis. And rRNA has two roles: initiation of protein synthesis and from the 5 prime end. And at the 3 prime end, it helps in signal side, initiation of mRNA. Now, when it comes to the site of ribosome, when both the units are attached only, it forms an active ribosome. Otherwise, the 3 prime and the larger subunit and the smaller subunit are going to be present in a free manner. So you have a peptidyl donor site, amino acid acceptor site, and an excitor site. This is for the necessary of production of your protein synthesis. Again, function of RNA or ribosomes is for protein synthesis. So two necessary components are your the ribosomal subunits tRNA having the anticodon, which is UAC, and mRNA molecule to be translated. So this is important. Just can I pause for a minute? I just want to check something. Before that, I hope DNA transcription was already dealt with you, in fact. So I'll just share an image which shows how the transcription machinery is going to, going to be there. Replication of DNA is one, and when it comes to DNA transcription, DNA transcription is wherein you have RNA polymerase is present having a 5 prime and a 3 prime cap, in fact, and then you get to see that um, production of the mRNA is being done in DNA transcription. Now, in translation, that mRNA, whatever is present, is going to be translated so that you are going to get functional proteins. So, protein synthesis is again of 3 types. We'll come to that. In this, you have initiation, and elongation and termination. So it is being done in both prokaryotes and eukaryotes, and it is always in the 5 prime to 3 prime direction. That is your amino end of the protein, of the mRNA to the carboxyl end, so that you have protein synthesis being done. So whenever units of ribosomes come together, that is when you have, in this ribosome itself, you have three types of what do you call it, uh, um, um, what do you call it, components present, which is your initiation codon. So the 50 subunit actually binds to the 30 subunit of mRNA. This forms the active ribosome or ribosome mRNA complex. This is attached by with the attachment of magnesium ions. This causes the attachment of the or formation of the active ribosome complex. So whenever there is a tRNA coming over, F-med tRNA, there is an mRNA easily or readily available. This causes the larger subunit to come. Already the smaller subunit is bound to the mRNA, and you have the initiation codon. You have the pool of tRNAs present, readily available. They will come, join over here. This causes the larger subunit of the ribosome to come, get itself attached to the mRNA and the smaller subunit forming the active mRNA complex. This again has the binding sites which are acceptor site, and then you have the P site, and then you have the what do you call it, the E site. So just a second. Whenever there is this, what do you call it, elongation that has to happen, the peptidyl site is filled with your N-formyl methionine in the case of prokaryotes, and then the amino acid site and the exit site are going to be empty. Next codon comes and goes and binds or is present in the amino acid site. This causes the direct binding of the amino acid tRNA. So you have different elongation factors also that are going to donate the amino acid tRNA to the ribosomes. So when this is the active ribosome complex present, first one you have your P site, or which is going to be filled. That is the peptidyl site, which is going to be filled with your anticodon tRNA having the respective initiation we are talking about. So AUG, so UASC anticodon has come, occupies the P site. Next acceptor site, it is going to go, and the next amino acid coding for on the mRNA will come and attach itself. Once this is done, then you have the production of methionine and valine amino acids. Because of which what is going to happen is the tRNA gets released after it goes into the exit site. And a formation of a dipeptide bond is taking place. In this manner, elongation is going to take place, and you have a transpeptidation reaction also, in which you have a peptidyl transferase, which is again located in the 50A subunit itself, which will help in formation of the peptide bond. So the alpha amino group that is present in the amino acid site A will go cause a bond with the alpha carboxyl group at the C terminal. So because of which water molecule is lost, because of which a peptide bond is formed. And you have a specific adenine base that is going to catalyze based on what kind of peptide is present, or what kind of so that respective adenine base will come and act upon. So you have the amino terminal, carboxyl terminal acting to form the peptide bond, because of which you have a growing peptide chain. So this is the 50A subunit, 30A subunit, mRNA. tRNA is coming along with the anticodons. This is in the 5 prime to 3 prime direction. This is in the 3 prime to 5 prime direction. So moment again is moment of this ribosome on the mRNA is known as translocation. So the peptidyl tRNA is going to move from the amino acid site or acceptor site to the P site, that is the peptidyl site. And along with this, one codon is moved along to the mRNA. So new codon comes, goes into the acceptor site of the ribosome. So again, your peptidyl site is empty. In this manner, it is going to be translocated. Again, for this to happen, you need energy. Energy is being given by GTP. So you can see over here, this is once the acceptor site is, the peptidyl site gets occupied, then there's a peptidyl transferase center bond being formed over here. This peptide bond causes or forms the amino acid, right? And then you have the peptidyl transferase enzyme acting on this. Once this codon gets transferred to the peptidyl site, and then the amino acid is formed, this is going to be empty. So it goes into the exit site, from which you have the deacylated tRNA being released from the exit site of the ribosome. So again, you have different kinds of factors that are going to be there. That is release factors basically. So you have initiation factors, IF1, 2, 3, elongation factors, and termination factors or release factors. So please give me a moment. I think I'll follow this. Right. Initiation codons in different pairs are have been given basically how they are going to occur. So pairing of the RNA with the mRNA is also being given over here. You can just have a look at it. Then you have different types of tRNAs that are present. So they can be F-met tRNA or met tRNA, and synthetase is added to it. So, and you have a synthetase and transformylase, which is going to give you either methionine tRNA or formyl methionine tRNA. What are the initiation factors now? When we see IF1, 2, 3 are the initiation factors. So these come 30S, they bind to the 30S ribosomal subunit. So whenever IF2 with bound with GTP is bound to the 30F, and along with that you have the tRNA also, F-met tRNA in case of prokaryotes. Along with on the mRNA, if they are bound, then it causes the production of the 30S initiation complex. This 30S initiation complex, after it gets formed, IF1, IF3 factors get released. And then you have the 50S subunit, which is going to be present. Of course, we are having water in abundance. So this causes release of IF2 and ATP is used. So GDP plus PI, because of which we are going to get the active mRNA initiation complex with the 50S subunit and the 30S subunit being joined together. And this is being assembled in the P site in the first place. Now, this is how initiation is going to take place. And elongation factor EFTU gets the correct mRNA correct amino acyl tRNA to the A site, that is the acceptor. Again, energy is used. So you have the EFTU GTP, which actually, whenever the GTP is present, it stops the esterification, or it stops that bond being attacked or damaged. So that is when this GTP, whatever is present, is going to get converted into GDP only when the proper codon is being present, and then the EFTU factor is going to be released into the cytosol again. Now, what do peptidyl transferases and peptide bond formation, what is going to happen? You have a 23S RNA and a 50S subunit present in fact. So, you have because of the proximity, meaning how near it is, all of them in one particular place and how they are oriented, because of which you have a peptide bond formed between the alpha, amino terminal and the alpha carboxy terminal. Now, elongation in the sense 50S is occupied with P site, and then you have A site empty. EFTU gets the other amino acid, that is your tRNA amino acid, and then it is going to go form a bond between the second three codons that are present, and then you have a peptide bond being formed with the help of a peptidyl transferase, and then all of this is being translocated with the help of elongation factors G. So again, release of energy through the form of GTP. GTP hydrolysis gives rise to GDP plus inorganic phosphate, and then dissociation of that particular tRNA from the exit site. This causes peptide bond to be formed. At the same time, the translocation of the active mRNA complex along the translocation of the ribosome along the mRNA. So this causes the peptide bond to elongate. And how is it going to be terminated? Release factors are present. RF1, 2, 3, like I told you, they recognize the stop codons. After all this translation, they occur, or whatever, the amino acid tRNA is going to come along the, anti-codons for the codons which are UAA, UAG or UGA. That is when RF1 UAA or UAG and RF2 UAA or UGA recognize the stop codons. This release factor recognizes that it is a stop codon, interacts with the peptidyl transferase that is present, which helps in adding the water. And what happens is, instead of recognizing the growing polypeptide chain, it recognizes the water. So GTP hydrolysis will occur, and if that because this will go bind to the EFG, elongation factor G, and the ribosomal release factor is going to release the ribosome. So in case of initiation, bacteria, that is prokaryotes, eukaryotes, are going to be different. So we have completed initiation, elongation, termination. Now let us just have a look at the differences in protein synthesis in bacteria and in eukaryotes. So FMET and MET tRNA, and it's a larger ribosome in the case of eukaryotes, smaller ribosome in the case of the prokaryotes. And initiation involves initiation factors and a cap recognition factor, that is IEIF 4E, and movement to this start codon AUG is through EIF2. And eukaryotic mRNA is usually circular in structure, because of which rebinding of the ribosomes is going to take place. And in elongation also, EF1 alpha, EF1 beta gamma are present. They're the counterparts of EF2 and EFTs basically. And bacterial EFG translocase is present in bacteria, whereas EF2 is present in the eukaryotes. Termination again, ERF1 release factor. It's the only one in eukaryotes. Whereas in the prokaryotes, you have EIF3, which actually prevents this premature ribosome reassembly whenever there is no initiation factor present. In eukaryotes, again, translation is through a large complex, and it is taking place inside the cytoskeleton, and that is what. And we have certain inhibitors of protein synthesis. Where are they going to act actually? You have antibiotics, bacterial antibiotics, in fact. So what is it that is happening over here? You have different kinds of antibiotics. Where are they going to go and act so that protein synthesis is not taking place? Streptomycin and aminoglycosides, initiation of that mRNA itself is not taking place. So whenever FMET mRNA initiation is stopped, you do not have bacterial protein synthesis occurring at all. Tetracycline binds to 30S subunit. So because of that, you do not have the binding of amino acyl tRNAs in the bacteria. Chloramphenicol stops the peptidyl transferase activity in the 50S ribosomal subunit. Cycloheximide, it stops the translocation in the eukaryote. So it's a poison for us. Erythromycin binds to the 50S subunit, inhibits the translocation. Uromycin in premature chain termination. So you have different kinds of proteins, or I think we have already seen. So regulation is by IRE, that is iron response elements. So this blocks the translation of the mRNA basically. So whenever there is high iron, it binds the iron. So you will not have the ferritin mRNA. So this ferritin is used to produce to store iron. Whenever there is low iron, so you have IREBP, it binds to the ferritin mRNA and blocks the ferritin production. So regulation through small RNA is also there. You have small interfering RNA and RISC and DISC in fact. So what happens is this is going to be cleaved, this, and if at all there is RNA interference proteins, because of which what happens is complementary mRNA is produced and degradation of that RNA is taking place. MicroRNAs again encoded through RNA precursor. You have again complementary mRNA being formed, and you have around 60% of the genes are regulated by microRNAs, which actually cause cancer and other diseases as well. So this is all about protein synthesis. I want you to just give me around 10 to 15 minutes of time. I just want to talk about gene regulation. So we have a certain, we have operons and the rest of them. At least half of it I want to finish in this particular class. So I request you to give me another 10 minutes at least. I'm going to stop share over here. Sure. So when we talk about gene expression and gene regulation, what is it about expression and gene regulation that we are looking at? Expression is, of course, DNA and then production of mRNA, and this mRNA is converted into the functional product, which is your protein. So you have small nuclear RNAs, non-coding proteins such as tRNAs that are present, which are used for translation, small nuclear RNAs that are present. And how are they going to be controlled? Through other genes, which are going to regulate. This gene expression is going to be regulated or controlled by different kinds of genes again. The whole concept is the whole activity should have a stop, start, regulation, how much is necessary. All of it is being done by genes itself. So in this, if you see, product of the regulatory genes is to initiate the turn on or turn off expression. So these regulatory genes, regulatory proteins, bind to DNA to either block or stimulate the transcription depending upon how they're going to interact with the RNA polymerase. So we have two types of gene expressions again. Constitutive, so that is extremely necessary, for the life to go on. They're known as housekeeping genes. What are they? Actin, then you have your dynein, some of them, and then you have your glyceraldehyde, 3-phosphate, GAP dehydrogenase. This is one of them. So many of them are housekeeping genes that is necessary for us to live. And then induced genes are for repression. So this again depends upon the response to the external signal. Suppose you eat a lot of sugar, there's a lot of glucose that is being produced in the blood. So your blood sugar levels are increased. So that is when the hormone insulin has to be produced, so that when insulin is produced, the insulin receptors are active and the insulin helps in the glucose molecules to go and get into the cell. And then this extra glucose, whatever is there, is being converted into glycogen. That is the next process. So these are the induction and the repression genes. So again, expression in the multicellular organisms can, most of the genes are present in the single cell itself. But proteins are again necessary. It can be in deficient levels or in extremely high amount of cell, because of which there is going to be an abnormality that is there. So gene expression causes an abnormality in the protein or in the product expression, because of which any kind of disorder can occur. So some genes are going to have higher expression level. Once they're activated, it's called as induced expression. Others will help in repression, or the expression of the product is going to be lower. It is called as a repressed expression. So we have exons coding sequences that are there. So exons are those that are you have your mRNA. This mRNA is going to get cut, and then it is going to cause the formation of the functional mRNA having only the exons. So pre-mRNA has both exons and introns. Introns is not going to be coded, but we are going to get the coding sequence with exons, which is going to give you the functional proteins. So if at all some exons have sequences that is not translated into the amino acids, so these untranslated regions are found upstream, that is before the protein coding sequence, and downstream, that is after the particular protein has been coded. So gene regulation mechanism is having a different kind of control over here. So different mechanisms are there for gene regulation, and it is more complex in eukaryotes than prokaryotes. That is because for us, we have different organelles that are performing different functions, which is not seen in prokaryotes. So this compartmentalization allows the specific function to perform without any kind of interference with the complete cell function happening. And you have a very good transcript processing, and regulation is at a distance because our cell is bigger than the prokaryotic cell. You have different organelles. So you have a cell and a tissue specific gene expression taking place, larger genome size, and the genes are scattered all through the genome. So different expressions can take place at a time. So what is happening during gene expression versus transcription? After this, you completely do not have translation occurring immediately as in the case of prokaryotes, but we have an RNA processing unit. Then RNA, so that's where you have your spliceosome complex coming in, you have your pre-mRNAs coming in, then you have your mRNAs coming. So that is one thing. And then you have your translation and protein processing. So how is it happening in the case of prokaryotes? In the prokaryotes, we have something called as the operon, which is a functionally related genes. They work together so that gene expression is being controlled in the prokaryotic cells. So all these genes that are going to control the expression of protein or functioning of the proteins are called as an operon. This operon has a promoting site which will start, if at all this is there, that means that yes, it is the binding site for an enzyme which is known as RNA polymerase. And you have an operator site wherein, this is going to be next to that of the binding site. So if operating site is free, and then that means that the protein synthesis will occur. If it is bound by a repressor, that means that that particular gene expression is not taking place. If gene expression is not taking place, protein synthesis is also stopped. And then you have the structural genes present, which after transcription translation encode to the different proteins, which are, for example, in the case of lac operon, this beta galactosidase permease transacetylase. So you have lac Z, lac Y, lac A present. The promoter sequence is a short sequence of DNA again, at which you have the RNA polymerase enzyme that comes, binds itself, attaches, starts the transcription. Now they are really recognized by RNA polymerase itself, through which when the binding occurs, transcription starts. Operator, again DNA sequences, and it is right next to your, your promoter sequence. So if it is blocked by a repressor protein, that means the transcription of the structural genes don't take place. Activators are again wherein if at all the presence of an activator is there, then the activity of RNA polymerases will increase. And whenever there is activator, the repression stops. So for example, when it comes to genetic code and protein synthesis, before the primary transcript, you have RNA and it is modified in many ways. So this is the cell, the chromosomes having the chromatid, DNA transcription with introns, exons. So introns are removed. So like whenever introns are removed after transcription, you have the post-transcriptional processing in which excision of the introns is taking place, splicing of the exons, and then pre-mRNA to mRNA. This mRNA protein synthesis will occur. That is translation. So gene modification, you have even addition of a 5 prime cap and a poly A tail. So this 5 prime cap, why is it necessary? So that this mRNA, whatever is formed, is not going to be cut down by different kinds of enzymes which are going to cleave the water hydrogen, water molecule bonds. And then translation can start for the ribosomes to come. So whenever at the 5 prime end you are going to add a 5 prime cap, that is guanine phosphate, which is being added over here at the 5 prime end, you're going to have a leader sequence also. This is the coding segment, and you have a stop codon here. Then after the stop codon, you have a poly A tail. So what is going to happen is you do not have other nucleases enzyme coming in and cleaving all this pre-mRNA present. So 5 prime cap poly A tail is one RNA modification, that is post-transcriptional modification that is being done. Addition of 5 prime cap and poly A tail. And then RNA transcription has taken place. Now translation. So translation, again you have whatever DNA is present, proteins, they are the link between the genotype and the phenotype, right? So this is the bridge between the DNA and the protein synthesis, that is the RNA. So during DNA transcription, strand is is going to be what do you call it, opened, and then mRNA, complementary mRNA is being produced, and then you have translation at the ribosomal level. So bacteria like the nucleus, so both transcription, translation are going to be coupled. So you have an mRNA where DNA transcription is still happening. At the same time, you have mRNA translation also occurring at the same time. Whereas for us, it is not like that. Because once the nuclear, what is happening is DNA transcription, post-transcriptional processes are taking place inside the nucleus. Translation is outside in the cytosol. And this is not going to change at all. DNA is going to give mRNA, it is going to give protein. So this is known as the central dogma of life. This will not change except in some viruses wherein, this mRNA will give rise to complementary DNA. So that is the only anomaly. Otherwise, always DNA transcription to mRNA will take place for the protein synthesis. And this is universal. This is why it is known as the central dogma of life. We have seen the genetic code as well. We have spoken about what happens in the genetic code. Transcription and processing of RNA also we have seen. And eukaryotes have different kinds of RNA polymerases 1, 2, 3. And you have promoter box, all of it taking place in the transcription. I will send this particular video. You can have a look at it. So this is what is all about gene expression. So when it comes to the operon concept, I think we have lac operon, tryptophan operon, histidine operon. So right now I'll talk about the, just give me a moment, pause share. I will finish up to the operons and then go ahead with what do you call it, the others in the next class. Right. When it comes to the lac operon, basic one, it is it was the uh, told about by Francis Jacob and Monod. It is seen in the prokaryotes, in fact. Right. So in this again, what we are going to see is it is going to code for enzymes involved in the degradation of a molecule which is lactose. So it's a disaccharide made up of units of glucose and galactose. In this, you have the like ZYA. You have the constitutional genes or the structural genes, and then you have the promoter and the operator. So in this, beta galactosidase breaks down the lactose into glucose and galactose. Lac Y is again is the galactose permease. This is the enzyme that is going to come, actively transports the lactose into the cells. And Lac A, you do not have. It codes for thiogalactoside transacetylase. So, and then you have a operator site, a promoter site, a repressor site that is present, and an inducer site also. So operator site is wherein you have the repressor like protein coming and binding, stops the protein synthesis. Promoter is where your RNA polymerase is binding, and repressor is wherein, whenever it is bound to the operator, stops the RNA polymerase from going forward so that protein synthesis or like transcription will not take place. So absence of lactose. If there is no lactose in the body, that is when the gene regulator is producing the repressor protein, goes and binds to the operator. So you do not have any kind of protein synthesis takes place. If there is lactose in the medium, that is when what is going to happen is whenever there is lactose present, then it is going to go, stops the repressor. Lactose goes binds to the repressor protein. So whenever repressor protein is not there, RNA polymerase comes over, binds at the promoter site, and protein synthesis will take place. And then the lactose, whatever is present, is going to be broken down by the different enzymes that are present. That is LAC-ZYA. So this is how the, what do you call it? And whenever the lactose molecules become less, then what happens? The functioning of this is not going to take place again, and the inducer will go because the lactose molecules are broken down over here, right? It goes, binds to the operator again so that the protein synthesis will stop, or I mean the transcription stops. Simple. So absence and presence of the lactose, in the medium is what is going to actually cause the bacterial, the transcription of the lac operon to stop or start. The next is we have positive regulation or catabolite repression. We will just have a look at it later. Tryptophan operon. In the TRP operon, I'll just have what is it that we are going to see? Tryptophan operon regulation of the transcription is necessary so that, whenever tryptophan is present, we are going to see coristinate, if it is present, formation of tryptophan inside the body. So it has a structural genes that are present and a regulatory region that is present. This regulatory region has the promoter, repressor, and operator and a leader sequence as well. So whenever this sequence is there, this is where we have the production of tryptophan which is taking place. In a gist, if you see, you have structural genes which are TRP E, D, C, B and A. E, D, C, B, A. So this encodes for the different kinds of proteins again, which are all anthranilate synthase 1 and 2. And then you have isomerases, And then you have tryptophan synthase B subunit, tryptophan synthase A subunit. So what is it that we're looking at? This has a following mechanism basically. Whenever there is tryptophan absent in the cell, then tryptophan, the repressor gene is going to, the repressor gene is going to encode, and that is actually, since there is no tryptophan, then transcription occurs. Chorismate is going to be converted by all of this, by transcription, translation, formation of tryptophan is taking place. So absence of tryptophan, then the inactive repressor is, the repressor gets inactivated, and then the promoter comes, binds to the promoter region, RNA polymerase comes and binds to the promoter region, operator region is present, and finally you have the production of the tryptophan. When there is high amount of tryptophan in the cell, what is going to happen? So we have a regulatory gene over here which comes over, and then you have the synthesis of the inactive repressor. So whenever the tryptophan is absent, this, what do you call it, the mRNA is synthesized, no RNA is being made over here. Then whenever the tryptophan is present, the active repressor comes and shuts down the gene, that is transcription. So whenever the tryptophan is high in the cell, it is being understood that it binds with the repressor protein. So conformation of that particular, the protein is taking place, and that goes and binds to the operator. So binding of the repressor to the operator protein. So what happens is the promoter is also getting overlapped. RNA polymerase cannot come to the promoter and cannot bind to the promoter region, because of which transcription is halted. And attenuation again occurs, in the case of bacteria, transcription, translation both are occurring at the same time. So as soon as the transcription is over, that part is being translated. So attenuation is found only in the bacteria but not in the eukaryotic cell. I will finish the tryptophan and then stop it over here because it is already 1:35. Histidine, arabinose, gene regulation, that is splicing, and role of chromatin will be dealt with tomorrow. So you have regions again in the case of the attenuation in the tryptophan operon, basically, wherein when you have different regions, you have regions 1, 2, 3, and 4, and they are complementary. So there's a loop formation over here. What happens is 1 and 3 are complementary to each other. I'm sorry again. It's like this. The 3 is complementary to both 2 and 4. 3 and 4 base with each other, form a loop-like structure which is known as the attenuator. This causes transcriptional termination. And then if at all there is 3 and 2 pairing, then no attenuation is taking place, because of which the transcription continues. So this is how the tryptophan operon works. 3-4 formation causes attenuation. If 1-2 formation or 2-3 formation is there, when the tryptophan levels are high, then 1 and 2 in the ORF is going to, 3 and 4 is forming a loop-like structure and termination of the protein synthesis. If tryptophan levels are low, then the ribosome pauses at the TRP codons in the sequence. So you have a paired structure or loop between the 2 and 3 sequences, because of which 3 is not available to form the attenuator structure, because of which TRP structural genes are going to be transcribed for the production of the mRNA. And when the tryptophan is low in the cell, that's what I said, the translation stops at the tryptophan codon. Loop between 2 and 3 forms, transcription is continued. So this is the feedback mechanism that we get to see when tryptophan is high, structural genes transcription not taking place. Tryptophan is absent or very low, transcription is continued, because 2 and 3 are forming the loop instead of 3 and 4. Chorismate is high, tryptophan transcription takes place. Chorismate is low, then tryptophan synthesis is not taking place. This is the feedback mechanism. Histidine and arabinose mechanism I will talk about in the next class. So this is about it for today. Yeah. Thank you so much.